This recording is for Biology 1406, Unit 5, the second lesson, Chapter 12 in the OpenStax textbook, Mendellian Genetics. So first we'll kind of take a look at what people were thinking about prior to the modern understanding of genetic inheritance. These are not necessarily held all by any particular group, but are representative of some of the ideas that are strongly held even today in individuals who don't really understand the biology and biochemistry of genetics. So one, the homunculus, which means little tiny man idea. That's that the gametes, and the only gametes that people prior to the modern age really saw, no one ever saw an egg, were, in humans anyway, were the sperm cells. And so the idea there is that there was a fully formed little miniature individual in there that would then mature inside the woman. The idea that like begets like, offspring are like their parents. And of course, there is some basis to this observation right here, but systematic observation shows that this is not exactly true. All the traits come from the egg, that females, in other words, controlled all the traits. Children would look the way they do because of characteristics of the female. In other societies, where the paternal heredity is a more important idea, the idea that males control all the traits. One little interesting thing about it that's true is males control the gender of the offspring. That is true, but only that one thing. Everything else is a shared interaction between both parents. The idea of blending inheritance, and that's the idea that offspring were inevitably the average of both parents. Well, if you take that to its extreme and keep doing that generation after generation, everybody's going to be the average, and we'll all look the same. Just basic observation shows that that's not the case. This one here that we, when we study evolution in Biology 1407, we'll recognize this as Lamarckian inheritance. Lamarck was a French biologist and evolutionary scientist who is quite talented, responsible for a lot of good stuff, but unfortunately also promulgated this idea, which turns out to be false. And that is changes happening to a parent, in the parent can somehow be encoded in whatever is passed off to the offspring, DNA we know now, and passed off to the offspring, all right? So a classic idea right here is that a blacksmith, would get very, very big, thick, heavily muscular arms because of spinning all day next to a fire, pounding a big, heavy hammer on on an anvil, forging things, or, working iron. Anyway, so the point is, it was well believed that a child would naturally be good at that physically. They'd have the physical characteristics for it because the parents had developed those characters. And of course, we recognize that the big arms and all the adaptations that happen in an individual as they take on a particular task is just that. It is adaptation in real time, but it is not heritable. And then this rather more arcane idea of pangenesis, and that there are, that all living organisms gather together these little particulate things that are gathered together and determine what an individual looks like when they're born. And there's a germ of truth in this, and of course the gemules we would recognize now as being genes, but that's not really, it's not quite the same thing. Anyway, this is just a little bit of historical context that adds a little bit of flavor, as it were, to the chapter. All right. So here's just an example of, this is a French text right here. Yeah, it says a Swedish text right here, but it's the French spelling. It's the French translation, perhaps, of the Swedish work. But anyway, yeah, the little tiny miniaturized person inside a sperm cell. Anyway, we know that that's not true. All right, the chapter is referred to as Mendellian genetics because of the work of Gregor Johann Mendel. As you can see right here, he lived from 1822 to 1884. And he was a monk in the Augustinian order. He had studied, along with religion, of course, philosophy and religion, he also studied botany and mathematics, and particularly a statistical version of mathematics while he was at university, along with preparing for the priesthood. So, he was a well-educated individual with a terrific amount of diverse knowledge that he accumulated during his schooling. And he is famous for working with garden pea plants. So a terrific amount, not all of his work, but some of his most important discoveries were made working with these plants right here. The pea is a legume. It is the plant that produces a bean pod that has very large round seeds that we eat as peas. Um, and what was useful about these plants is that he noticed that a lot of the plants that he was growing in the pea garden, their garden, growing their own food, very, very typical back in the 1800s in a monastery. Those plants had dichotomous traits, which means that, plants in a population either had one character or a different character. That's what dichotomous means, one of two choices, tall or short, curly or smooth, whatever. And there were characters that had to do with color flower, white versus purple, seed shape, seed color, and all kinds of things like that. Also, he observed that many of those characteristics were true breeding. Now, this is something that lots of people were attempting during this time, is to, particularly in the flower, the business of growing exotic flowers for people who could afford to pay for them. And that is growing true breeding flowers, flowers whose offspring, the seeds, and therefore the young that are grown from them, look just like the parent. That's what's meant by true breeding. A useful characteristic for the peas that he worked with is that they are monoecious. And that means the flowers are both male and female. In animals, we would refer to that as hermaphroditic. They have both, sets of reproductive organs at the same time. All right, and so it makes it easy to pollinate one flower from another one and vice versa. And also, of course, when he collected the peas, the seeds, each pea was a new individual, and so because they were relatively large and handy sized seeds, easy to manipulate and work with, and also they grew up quite quickly. So a whole bunch of interesting characteristics. Because he chose these plants, he turned out to be able to do a terrific amount of excellent work in a relatively short period of time. All right, so this is the notation that he worked with. So all of his notes will be along in these lines right here, and terminology. It would have been in Latin in his work, because everybody wrote in Latin at the time in the sciences. But if translated into English, this would be it right here. So the parental generation, that's fairly obvious. P, parental, true breeding parent plants. And so he only worked with true breeders, plants that had white flowers, and their offspring only had white flowers. And when you bred white flowers to white flowers, you got white flowers and all that kind of stuff. F1, the first filial generation. So if you breed two parents together, the offspring from that first breeding is the F1 generation. And then if you take siblings, brothers and sisters from that F1 generation, and breed them back to themselves again, so inbreeding basically for pea plants, that's the F2 generation. So there's a first generation, a second generation, and a third generation to produce the data that we are going to talk about. And you could keep on going for F3, F4, F5, but we'll only talk about these three, P, F1, and F2. Siblings, brothers and sisters, cross sibs. That is a cross between a brother and a sister. In other words, a female flower and a male flower right here to produce offspring. Inbreeding is what we would call that if we were talking about humans. But these are plants, so we don't care. A back cross, where you take an offspring and breed it back to a parent or another ancestor, and a self cross. Some plants can do this, where you can breed a plant to itself. So those monoecious flowers. In some plants, the female part of the flower can fertilize the male part of the same flower or another flower on the same plant. Some plants do this, most don't, but it is a thing. All right, and then true breeding right here, meaning parents whose offspring always have the same traits. So they have the same traits. And what we would recognize today is that true breeders are genetically homogeneous. They only have one particular genetic version of a genetic trait in their genome. All right, so here's how it works. True breeding plants, and this is a formal cross to test for a trait right here. So we have what we think are true breeding purple flowers right here, and another plant that we believe is true breeding, and we cross them with each other, and they produce a whole bunch of seeds, and we take a look at those offspring, and the offspring are all going to be purple right here. All right, and then we mate them back. We cross these to each other. So we mate siblings to themselves, and take a look at the second generation of the F2 generation. This is the parental generation, but the first generation of offspring, the second generation of offspring. Anyway, true breeding, all the same color, long and short. Now, this is where it got interesting. He worked with true breeding white peas that had been proven to be true breeding, just like this. White times white equals white. White siblings mated with each other produced white offspring, etc., etc. So he had that, and he had purple ones. And then he took known true breeding purple plants and known true breeding white flowered plants of these peas and crossed them to each other, and all the offspring were purple. No blending. All the offspring had the trait of just one parent. And then he took two individuals of these, or actually many pairs of them, but the idea was to mate siblings back to each other. And what he got was, if you took a look at, say, 100 offspring flowers, large numbers are required to get these sort of average statistics right here. But if you looked at enough flowers, 100 offspring or 1,000 offspring, you would get pretty consistently 3/4 that had the purple coloration, but 1/4 that had the white coloration of one of their grandparents. And if you repeat the experiment, you get the same sort of thing. Now, it's not perfect. It's not exactly 3-to-1. It might be 780 to 220 one generation, one time in the experiment. But the numbers keep orbiting around this 3-to-1, 750 to 250, 75 to 25, depending on the sample size. Consistently, no matter how many times, you got this sort of pattern right here. All right, and that was very, very interesting. What the heck is going on? All right. So here we're looking at it, looking at the larger flowers. So, purple and white crossed to each other. All the offspring are going to be purple. You cross these individuals back to each other, and you get individuals, we're about 3/4 of them have this color right here, and 3/4 of them have this color right here. And this is where we come up with the dominant and recessive terminology. Dominant because this is the more dominant color, always more abundant in the F2 generation, and the recessive trait less frequent in the F2 generation. That's where the dominant versus recessive comes from these, this 3-to-1 ratio right here, kind of consistently. All right. And so he did that for not just flower color, but seed shape, seed color, height of plant, any number of traits that were dichotomous on these pea plants. And he found that lots of them obeyed these same rules. All right, a tall plant, true breeding tall plants, true breeding short plants. So then he'd have true breeding tall plants, true breeding short plants, and cross them together, and all the offspring would look just like one of the parents, let's say tall, all of them tall. And then we cross those to each other again, and we get 3/4 tall and 1/4 short. I don't know if it works that way for I have to check. It might be 3/4 tall, short, and 1/4 tall, short being the dominant and tall being the recessive. It just depends. How do we know which is dominant and recessive? It's not the biggest, strongest, and fastest. It's the most abundant versus the rarer. That's what defines dominant versus recessive. All right. So that's what we were looking at right here. So we know now that we've got this dominant sort of character that appears in a ratio of typically 3-to-1, and this recessive character that appears, consistently as 1 versus 3 of the other character. All right. So what this tells us is that, whatever is being handed off from the parents to the F1 generation, and importantly from the F1 generation to the F2 generation, is, there's some particle. So there's a white particle coming from white flowers that in some way, shows up, not in the F1 generation, but in the F2 generation, intact, unchanged. And that's the idea of particulate inheritance right here. Now we know that the particles are in fact genes, or more specifically, versions of genes. The word is allele. All right. So Mendel didn't know what was going on right here. All right. biochemically. He didn't know the chemistry underlying it, but he was gathering very, very careful numerical data on his plant breeding experiments and creating mathematical rules that seemed to drive inheritance. That's the work he did. Now, these days, we'll recognize, that what he was working with were these things right here. So a trait is just the name for something that you can see, a characteristic of an organism. There's also traits that you can't see, they're internal, like what version of a digestive enzyme do I produce? How big are my bones going to be inside? All kinds of things. But if they're genetically controlled, they're called traits. So in other words, if there's a genetic component and there's a heritable component to it, it is a heritable trait, and that's what we're interested in here. And lots of them can be seen, so we call those phenotypic traits. Phenotype means something that you can see and look at. So the different traits, the different versions of traits, do we have blue eyes versus brown eyes or something like that? That's due to different alleles for the gene controlling the trait. So a different allele is a different version of a gene. You get a biology textbook, but you might get the Campbell textbook, or you might get the OpenStax textbook, or you might get the McGraw-Hill textbook. Those are all versions of the same text, talking about the same subject, and that's kind of like alleles are to genes. Alleles are specific versions of a more general character called a gene that controls some trait. And then, of course, what Mendel was uncovering was this relationship between alleles where there was a dominant allele and a recessive allele. The genotype is the genetic basis for what we see, and that's important when we look at the F1 generation versus the F2, and we'll get there in a moment. All right, but so a genotype, there are two different possibilities of genotypes for given alleles for a given gene that codes for a trait. And that is a homozygotic genotype, which means both alleles are the same. They code for the same thing, blue eyes, both of them. And then heterozygotic, that's another possible genotype for an individual, where the individual has two different alleles for the same trait. Notice right here we've said big P and little p to represent one version, one allele for P color and a different allele for P color. Traditionally, the uppercase P is purple, the lowercase p is white. Notice up here when we had said homozygote, we've got both either the big P or both the little p. So homozygous for purple or homozygous for white. Well, that's a true breeding plant right there. This one is a true breeding plant if that's its genotype, and this is a true breeding plant here if it's its genotype. And remember, we're talking about sexually reproducing organisms that we looked at in the last chapter. And pea plants, like people, are diploid organisms. So each cell in every cell of that plant has two copies of each chromosome, and therefore two copies of the gene coding for each trait. And they can be homozygotic, pure for a particular trait, or they can be heterozygotic, different for a particular trait, two different versions of that allele right there. All right. And a zygote, of course, is the fertilized egg that is the product of fertilization. So these plants, just like people, ultimately produce haploid cells called sperm and eggs. The sperm are housed within the pollen grain. The egg is retained within the female part of the flower. And eventually that pollen grain releases a sperm cell that fertilizes an egg. And you get the fertilized zygote, and that's going to lead to the embryo, the new pea plant within the seed, the pea itself. All right. Some other useful terms. Gene, a specific location on some DNA, a particular location that encodes information causing a trait. We looked at gene expression regulation in the last unit, so hopefully you've got a good idea about what we mean by a gene. It's a distinct bit of DNA somewhere on a chromosome. And a locus is that location where a gene sits. Alleles, versions of genes, reciprocal cross right here. This should say formal, but I'll fix that later. Mating cross, where a previous cross is repeated with the parents exhibiting versions of phenotypic or reversed by sex. So male white flower versus female purple flower. And then the reciprocal cross is male white flower, female purple flower, etc., etc. How do they get that? If you cut off the male parts of the flower, all that's left is a female flower. Mendel did this. If you cut the tip off the female part of the flower, all you've got left is a male flower. So you can control gender of reproduction and all of that stuff as well, simply by manipulating the flowers. Wild type allele, what you most often see in the population. The wild type allele for color of flowers on sunflowers is most definitely yellow. I've never seen a mutant allele as far as color in a sunflower, but if they're out there, you're going to have a sunflower that's a different color. And if we've got a genetic basis for that different color, that's what we refer to as a mutant allele. Non-normal and typically rare. The wild type is usually quite common in a population, and the mutant is relatively rare in the population because oftentimes it's not that old. It didn't happen so long ago. All right. So here let's look at a monohybrid cross, which is the proper name for breeding individuals together while looking at one trait. One trait. Mono, it means one, and this is one trait, so flower color. So we looked at this before, but now let's add a little bit more detail to it. So we've got an individual that is homozygous. It's a pure breeding purple flower. So genetically, it has both copies of the gene that codes for purple, which by convention is uppercase right here. It produces gametes. Well, gametes only have one copy of the gene, one allele, so it'll just be a single P. Same thing for true breeding white plant right here. It's homozygous for the white alleles, lowercase right here, and the gametes it produces are going to be they're just going to have a single copy of the lowercase of the recessive allele here, with the lowercase letter. And when you breed them to each other, you get offspring that are necessarily heterozygotic. That's the only possibility, because this plant can only make gametes that have the purple allele. This plant can only make gametes that have the white allele. And so the offspring must have one of each of the alleles. That's the only possibility. That's the only way that would work. So all of the offspring are going to be heterozygotic in this F1 generation right here. And we know that purple is more is dominant in expression over white. And so we go ahead and symbolize it here. Then when we cross these guys to each other and we do enough of these simultaneously that we get a big set of offspring, so that random errors and random death of seeds and stuff like that doesn't skew our results, we see that we pretty consistently get this 3-to-1 ratio right here. All right. Whoops. But we'll take a look at it in a bit. It's called a Punnett square. There's a tool to help kind of decipher what's going on right here. Remember, this heterozygote, when it produces gametes, it can produce one of two types of gametes. A gamete that either has the purple allele or a gamete that has the white allele. It can make 50% of each of those. If it makes 1,000 pollen grains, 500 of them statistically will carry the purple allele, and 500 of them will carry the white allele. Same thing for this heterozygote over here. Remember, these are siblings of the cross between these guys right here. And so what we end up getting is when we put those gametes together, 25% of the possible combinations from this and this are going to be both of the dominant alleles right here. Another 25%, the dominant allele here from the recessive allele here. Another 25% of the population, the recessive allele here and the dominant allele from this one right here. So it's kind of an inverse right here. We always write the dominant first and the recessive second. And then finally a 25% just like this of getting the recessive alleles, two of them together. So genotypically we've got a 25% chance of being homozygous for one allele, the purple, 25% chance for being heterozygous for the purple allele, 25% chance of being heterozygous the other way around, dominant from this one, recessive from this, as opposed to dominant from this and recessive from this, which is what this one is. Added together, the difference is trivial ultimately. So 50% of the offspring will look like this or be genetically like this, and then 25% due to recessive times recessive right here. So genetically we get 1 to 2 to 1. Homozygous for purple, heterozygous, we don't need to say anything else, and then homozygous for white, 1 to 2 to 1, or 25% to 50% to 25%. Phenotypically, what we see though is this. Individuals that are homozygous for purple look purple. Individuals that are heterozygous for purple still look purple, just like the F1 generation up here. And then individuals that are homozygous for the recessive allele, only they take on the recessive character white. And so there's the genetic basis for dominant versus recessive. A dominant gene, one copy of it or two give us the dominant effect. A recessive gene or a recessive allele, properly, it takes two copies of the recessive allele to get the recessive character. All right. So Mendel's first law, let's just read through it. Heredity is particular. That's these intact things that whose character doesn't change. A particle, now we know, is a gene that codes for white, or a particle that codes for purple. All right. From his mathematics, it was clear that each group breeding parent carries two of those hereditary particles for a particular trait, flower color. All right. Those particles come apart during gamete formation, so they travel individually in gametes, not together as pairs. And then when you mate individuals together, those particles are back recombined again, but randomly, clearly randomly. From this mating right here, we get all three genetic possibilities of combining what's carried by these two individuals right here. And again, this one is doubly common because there's two different ways to come up with this recipe. This from the first parent, this from the second one, this from the second parent, this from the first one. And so that's the Mendel's first law of inheritance. To test his hypothesis, so he made this hypothetical explanation for what was going on, and then he generated something called a test cross that could prove whether this was the case or not. So let's look at a test cross. All right, so here's the test cross that he did. And the way it worked is he took a pure breeding purple flower and a pure breeding white flower, just like we looked at before, and bred those together so that he got these purple offspring. All right. And then he bred that purple offspring of these to another purebred white flower right here. And the result, when he did this a whole bunch and collected lots of seeds, was real consistently a 50-50 ratio of purple flowers to white flowers. What this tells us now is that the gametes that are being produced here, they're all white gametes. That's the only thing that can be produced. But this plant's producing gametes that are half white, and half of the gametes code for the white. They're the recessive lowercase p, and the other half of the gametes must be coding for the uppercase dominant color, purple. And of course, recessive times dominant gives you purple, and recessive times recessive, because this is a heterozygotic individual, born of two different parents, gives us the recessive character right here. And so consistently, if you do this test cross with a heterozygote mated to or bred or crossed with a recessive homozygotic individual, you'll always get this 50-50 ratio. So if you don't know the genotype of this to start with, once you've done this test cross and you get this, you now know the genotype of this thing. You always knew this, 2 copies of the recessive allele. So that's a test cross. If you got 3-to-1 in the results, then you'd know that this individual here was homozygous for the dominant allele. And that's what a test cross is useful for. Work with a recessive plant. You know what its genetics are by looking at it. And a plant that you hope is dominant or is homozygous, pure breeding for the character you want to make a lot of money off of, test it against this and look for the genetic rate, the ratio, the phenotypic ratio of offspring. All right, so here's the Punnett square way of solving these problems. It's this is just a diagrammatic way of generating this right here, or this right here, or that right there, or this right here, okay? Punnett square is just a nifty tool. So let's go ahead and take a look at the Punnett square. So you've got an individual right here that's homozygous dominant and an individual that's homozygous recessive. So you track out, just to be tedious, we'll go ahead and make 2 columns right here. So all of the gametes either have the dominant allele or they have the other dominant allele. And same thing for the gametes from the other flower right here. All of them have either this recessive allele or the other recessive allele, so this column right here. And when we combine them together, of course, all of them are going to be heterozygotic. This times this is heterozygotic. This times this is heterozygote. And there's your F1 generation that we looked at a while ago. This F1 generation right there. All of them are heterozygotic. Our Punnett square makes that really, really clear. All right. Then we take two of those F1 sibs and cross them to each other. Remember, each F1 sibling has two possible alleles that it can donate to a gamete. So we have one column right here for the dominant allele, one column for the recessive allele from this purple plant, and same thing over here from this purple plant right here. And then we cross them. And so we've got dominant and dominant, dominant and recessive, another form of dominant and recessive, and then finally recessive and recessive. 25% dominant homozygous, 50% heterozygote, 25% recessive homozygotic. You could do the same thing with seed color. Green seed, yellow seed, but we don't know if it's green is recessive because previously we got the 3-to-1 ratio on seed coloration, with seeds being typically showing up in 3 times as abundantly yellow seeds as green seeds. So we know yellow is dominant, but we get this yellow seed and we know it can have one of two genotypes. It can be heterozygotic or it can be homozygotic for the dominant allele, uppercase Y here for yellow. And so when we cross them, if it was homozygotic for the dominant allele right here, all the offspring would be yellow. The F1 generation would all be yellow. But if it was heterozygotic for seed color, the offspring would be 50% yellow and 50% green. Test cross. All right. So here's just doing that same sort of work right here with seed coloration. This is a monohybrid cross, so two purebred strains, pure breeding green seed plants, pure breeding yellow seed plants. The F1 generation are all yellow seeded, so that means yellow is dominant. And then we do the F2 generation and cross them back to each other, and we get this classic 3-to-1 phenotypic ratio of yellow flowers to green flowers. That's the classic ratio we're looking at, which tells us yellow is dominant and green is recessive. And then also note, genotypically, we have 3 different genotypes, 1 to 2 to 1 of the genotypes. 1 part, or 25%, homozygous for the dominant allele, 50% heterozygous, 1 of each allele, and 25% homozygous for the recessive allele, 1 to 2 to 1. Phenotypic ratio is 3 to 1 for the coloration. All right, now I'm going to skip ahead a bit. We'll come back to probability in a minute because it is important, but we're going to leave it for a second and show you why you need to learn probability. And we're going to leave the pedigrees behind for a little bit as well. And now we're going to look at a dihybrid cross. So we looked at a monohybrid cross before. A dihybrid cross tracks the same sort of thing, but for 2 characters, dihybrid. So if we were looking at 3 characters, that would be a trihybrid, etc., 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 etc. And Mendel did dihybrid crosses, and in fact he did trihybrid crosses. The collecting data becomes increasingly difficult as you carry this on. But let's take a look at a dihybrid cross. You can still do a Punnett square for this pretty quickly and gather the information pretty rapidly. All right. So we do the same sort of thing. We have pure breeding strains, for whatever characters we're looking at, but now we're looking at 2 characters. And just to keep it neat and tidy, one plant is dominant for 2 characteristics right here. The coating of the seed, which is characterized by the R right here. R is used to symbolize it. And so that's a smooth seed, R, and Y for yellow. So smooth yellow seed is dominant, right? That's most commonly seen in these particular pea plants. And then there's another variety that is green and wrinkly seeds. And these are pure breeding plants as well. The parental generation must be pure breeding. So, crossing them to each other all the time just gets you nothing but green, wrinkly, and crossing these to each other all the time gets you yellow and smooth. We don't care about other things like flower colors or stem height or stuff like that. That can be whatever. We're only paying attention to one physical, or now two physical characters. So we breed them to each other, and just like before, we're going to get individuals that are heterozygotic, but now it's heterozygotic for both characters. Heterozygote for flower coloration and for seed coloration, and heterozygotic for seed coat, rough or smooth. And so they look yellow with smooth seeds because those are the dominant, or smooth coat, because those are the dominant characteristics. But we know genetically that the F1 generation must be heterozygotic for both characters. Then we go ahead and look at the gametes they can produce. So remember, one of each of these characters can be introduced into the gametes right here. One of each character, but not both of a particular character. So we can get one character for seed color and one character for seed coat. So here we've got dominant and dominant, so yellow and smooth. Here we've got recessive and dominant, so green and smooth, and then the reverse right here, yellow and rough, green and rough. And then we do the same thing down this side right here. So because we've got two characters, now we've got four distinct gametes we can produce. Remember, we're just looking at two characters right here. Remember when I talked to you in meiosis about random assortment of chromosomes across the metaphase plate in meiosis, 2 to the 23rd, say 23 traits, but 23 chromosomes in that particular case, we would have 8 million and something, 8,300,000 and something odd things we'd have to put in a row right here. So that would be monstrously and huge. We won't even bother with that, obviously. But we could do a dihybrid cross, and then we cross them. It takes some time, but you pound your way through it right here. And what we'll find is we get a 3-to-1 ratio. We get a 9-to-3 to 3-to-1 ratio of the combined characters right here. And then if we look at just a particular character, notice that this is a 4x4 grid. 4 of these things are green. Very clearly 16 of these things, 4, 8, 12. 12 of these things are going to be yellow. So it's 3-to-1 for green versus yellow versus green. And if we look carefully at which ones are wrinkly and which ones are not, we'll find that it is 3-to-1 smooth versus wrinkly. But the combinations are important right here. And Mendel got these ratios consistently. 9, both dominant, 3 dominant for one character and recessive for the other. Another 3 that were the inverse, recessive for the first character, but dominant for the second character. And then finally 1 out of 16 that were recessive for both characters, 9-to-3 to 3-to-1. Phenotypic ratios, what we can see. These are all the genotypic ratios. They're all buried in here. We won't bother counting them up right here. There is a pattern there as well. Tellingly, there's only one way in which you get homozygous dominant for both characters, and only one way you get homozygous recessive for both characters. So the phenotypic pattern goes 1 to 2 to 3 to 6 to 3 to 2 to 1, across. You can work it out when you dig into the Punnett square, and all those numbers should add up to 16, which is a 4x4 grid. Anyway, 9-to-3 to 3-to-1 is the Mendellian ratio of phenotypic characters right here. What this tells us, all right, so this is what's come to be known as Mendel's second law, and that is the law of independent segregation. So genes coding for characters, color of seed, shape of seed, can function independently of each other. What happens to one doesn't in any way influence what happens to the other one. That's what's meant by the law of independent segregation. We now recognize that what's happening right here is that the gene coding for seed color is on one chromosome, and the seed coding the gene coding for the shape of the seed, smooth versus rough, is traveling on another chromosome. That's what's meant by law of independent segregation. And to see if that's true, If you do a dihybrid cross with pure breeding strains right here, you'll consistently get a 9:3 to 3:1 ratio of offspring if you have enough of these. If you just do 16 seeds, random error, is going to mess these values up. But if you do a big pile of them, 100 of them, or 1,000 of them, or 10,000 of them, you'll increasingly home in consistently on this 9:3 to 3:1 ratio. So that's a dihybrid cross, telling us that in this particular case, these two characters, seed color and seed shape, travel independently of each other. All right, so here's just another dihybrid cross that's been run. I won't go ahead and talk about it, but it's an example. All right. Now, just to make things complicated, the relationship between alleles isn't all just dominant and recessive. Mendel hit some of these other things as well, but he focused most of his work on where he found dominant versus recessive relationships between alleles. But here's a snapdragon. It's a flower. It can come in one of two colors. Well, actually, one of three colors. They can be red, they can be pink, or they can be white. All right. And what's happening right there is that if you get two copies of the red allele, you get a red flower. If you get two copies of the white allele, you get a white flower. But if you would get one copy of red and one copy of white, you get a mixed effect, and that's the pink flower right here. So that is an example of incomplete dominance. So if you breed in snapdragons, if you do the monohybrid cross and take pure breeding red flowers and pure breeding white flowers, all the offspring will be pink. All of them, all the F1s will be pink. Then when you cross them back to each other, you get this, which you'll recognize is a genotypic ratio. 1 part homozygous for one character, 2 parts heterozygotic, 1 part homozygotic for the other character, or 25%, 50%, 25% for a total of 100%. But now, the genotypic character is also obviously the phenotypic character, and that's what's meant by incomplete dominance. Quite a few genetic characters work that way. Now, another interesting thing, we can have more than one allele, more than 2 alleles, even more than 3 alleles for a particular trait. So here we've got multiple alleles for a single particular trait. So what we're looking at right here is just simply 3 different alleles that have a different phenotypic effect. And so here's a purebred individual for this allele, pure here, pure here, pure here. And these are 4 different alleles that we can find in these designer rabbits right here. Wild type brown fur, most common. And then chinchilla, which is black tipped white fur. So it gets you a very interesting kind of color right here. And then the Himalayan. As we can see right here, generally white coloration, but all the distal parts of the body, tips of the legs, tip of the tail, tip of the nose, tip of the ears, black. And then albino, just white fur right here. And so what we're showing is that we've got 4 different alleles, all of which can get different combinations. No individual, of course, can have more than 2 of these alleles. So we can combine brown and Himalayan, or brown and chinchilla, or chinchilla and Himalayan, etc., etc. We've got lots of possibilities, that we can combine together. No individual gets more than 2 copies, though, or 2 alleles, even if it's 2 different ones. In these cases, they're all homozygous. All right. Some other ways in which genes work. We know that this is true now, and that's something called homeotic. These are homeotic mutants right here, but homeotic genes. And these are genes that control a whole bunch of characters, because what they are is genes that control whether a whole bunch of other genes get expressed or not. And so that's another way in which genes express themselves. So there are these things called homeotic genes. A mutant is just a malfunctioning one, which is how we discovered they existed. Here's a fruit fly that looks normal. It's got its normal short little antennas with a bristly little hair sticking off of it. And this fly over here has these great big stubby malformed antennae. Flies have very short antennae, unlike a lot of other insects. So if you see something that looks like a fly, but it's got long antennas, it's not a fly. It's something else. Anyway, so a little bit about that. So we're not going to go into any details here, but recognize that, another way in which genes can express themselves is by controlling many, many other characteristics at the same time. All right. I'm I'm not going to bother with this one right here. We'll just move on past this one. All right, sex-linked traits. This is really, really important, and one that you're going to need to know. So we discovered this by looking at fruit flies right here. And we've got fruit flies, different genders and different genotypes for things like body color, terminal body color right here, and the color of the eyes right here. So notice we've got red right here, but then we've got brown on both of these individuals here. And, you know, a bunch of different things going on. Eye color, actually we've got more. Cinnabar, sepia, vermilion, white, and red. So that would be vermilion and that would be red right there. There's white right there. So a number of different genes. Okay. Red eye color is the wild type and dominant to white eye colors we can see right here. Okay. Interestingly, the gene for eye color is on the X chromosome in Drosophila, these fruit flies right here. So what does it mean when there's something that's sex-linked? The X chromosome, females carry an X chromosome, 2 of them. Males only carry 1, and they carry a Y chromosome. So this is a group of insects whose gender determination is the same as for same as for mammals, where it is homozygous for X chromosomes makes you a female, being heterozygotic for the sex chromosomes, having 1 X and 1 Y chromosome makes you male. All right, well, the eye color gene only exists on the X chromosome. The Y chromosome is famously really small and doesn't have genes coding for a whole lot of other stuff except how to turn a female into a male, which is the way it works. And so the Y chromosome doesn't carry any information about eye color. So this makes inheritance a little bit interesting. So here we've got a fly with white eyes, and in the previous panel we noted that white eyes are red eyes are dominant to white eyes. That means white eyes are recessive. So this individual with white eyes right here we know is genetically recessive. It's got 2 copies of the white allele on its X chromosomes. We have to indicate when we want to do a sex-linked inheritance question, we must pay attention to the sex chromosomes themselves as well as the allele for a gene that we're interested in. And the male gets 1 X chromosome. It's got red eyes, so it must have the dominant version, capital W in this case, as opposed to lowercase w over here. And then we breed them to each other. So the male can make an X chromosome carrying the eye color gene on it, or it can make a Y chromosome carrying no eye color information. The female can only produce eggs that carry X chromosomes because that's all she has to offer. All the eggs have X's in them. And in this case, all the X chromosomes, because she's homozygotic for white eyes, carry the gene for white eyes. Remember I told you in humans, the male controls gender of offspring. Well, in fruit flies, it's the same thing. The female can only contribute to making further female flies. The male can contribute to making a female fly or a male fly. So we do the cross right here. And so these 2 flies are male by definition because Y times X is male and Y times X is male. And since these carry no eye color information, the only thing of interest is the eye color information coming from the eggs, and that is white eyes. So these flies have white eyes with only 1 copy of the white allele. They're males with white eyes, but they only have 1 copy. It's a recessive allele, but now only 1 copy is creating the recessive trait because it's on the male animal that is heterochromatic for its gender, X and Y system. There are others out there, by the way. Most insects use something else. A lot of vertebrates use something else, but mammals and fruit flies in this particular case, and I assume by extension all flies, use the XY system. And so these females right here are heterozygotic for eye color, and red is dominant, so that's what we see. So we get 50% heterozygotic female and 50% homozygotic recessive male. And that's a sex-linked characteristic. Here we're going to look at it in humans, same sort of idea right here. We've got a recessive trait. It could be color blindness, which is also carried on the X chromosome and affects humans, red-blue color blindness. It's an X chromosome trait. So we can have a father who has perfectly fine color vision. He's got 1 X chromosome and the gene coding for his rods and cones is working fine right here. The woman also has perfectly fine vision, but it turns out she's heterozygotic, and she's got the normal gene that gives her full color vision, but on her other X chromosome, she's got the recessive malfunctioning gene that causes color blindness. In this particular case, recessive is no function. The gene doesn't make anything happen. So he can either determine, that an offspring is male or female by donating an X or a Y chromosome, and only those future females get some information about color vision from him because it's only carried on the X chromosome. And individuals getting his Y chromosome are going to be males. So here they are, male and male right here. She has a split. 50% of her eggs can be carrying the normal allele for color vision and 50% the recessive. So what we end up right here is a male with normal vision, a female, so 25%. If they had 100 children, we would get this sort of ratio. If they only have 4, there's a lot, the odds they could all be this way, or they could all be this way, or they could all be this way. But a big set will inevitably get this natural ratio. 25% of them are going to have, boys with the normal vision. 25% of the girls are having normal vision, and it turns out are genetically homozygous. Another 25% of the children are girls and still have normal vision, but they're carriers for the recessive allele. It doesn't function on them. The dominant allele, dominates, so they have color vision, but they carry the recessive allele. And 25% of the boys right here carry the recessive allele and therefore must be colorblind. And so color blindness predominantly affects males in the population. It can still affect females, but they've got to have 2 copies of the recessive allele. We already know the genetics for that. But on sex chromosomes, it gets a little more complicated. And so statistically, boys are 25 times more likely to show up with color blindness than girls. Hemophilia, free bleeding, is another one that's on the X chromosome. And so again, males are 25% more likely to be hemophiliacs than females from a given lineage of parents. Now, a few other things. There are such things as lethal alleles. So if you get a copy of this lethal allele, you die as an embryo and you're not born. So in this case, the results look a little bit odd. So, if you've got 2 copies of the lethal allele, you're dead if it's recessive. And so if you cross individuals that are carriers for the lethal allele, what you'll end up getting is 33% of them are going to be homozygotic for the normal character, and 66% of them, twice as many, are going to be heterozygotic, and that's it. You've just got, because the other 25% went away, all you've got left is 75%. And so if we just reduce that down, as those 3 possibilities being everything, that's why I did 33 and 66, adds up to 100 roughly. But you'll get, 1 part heterozygotic normal and 2 parts heterozygotic, heterozygotic. And you don't see the homozygous recessives because they don't live, they're gone. That's what we mean by a lethal allele. And so, the offspring outcomes are a little different, and you have to know what you're looking for right there. Now, what if we want to look at more characters? Well, we could do a Punnett square, but it gets really tedious really quickly. But if we just use some, organizing right here, some nice tabular work right here, we can take a look at seed color, seed shape, and whether the plant is tall or short. And remember, I was trying to remember that previously for, say, a pea plant. It turns out tall is the dominant character. And so 3 times 3 times 3 will get 27 round yellow tall plants, and 9 round yellow dwarf, and 9 yellow wrinkle tall, and so on and so on and so forth. You can go ahead and run these numbers right here. And there is a characteristic phenotypic ratio for a trihybrid cross that, as you can see, it's 27 to 9 to 9 to 3 to 9 to 3 to 3 to 1 consistently for 3 characters, as long as they're each on a different chromosome. Now, something for meiosis that makes this a bit messy is when 2 different characters genes for two different characters are actually riding on the same chromosome but get separated due to crossing over. So if there's no crossing over and the alleles are not separated, let's say these A and B alleles right here, and you do a dihybrid cross looking at those traits, it'll look like a monohybrid cross. You'll end up with 3/4 that have both dominant traits and 1/4 that has the one recessive trait. It'll look just like a monohybrid cross, except for both characters at the same time, if the genes are on the same chromosomes and don't get separated. But if they get separated by crossing over, like we're seeing right here, we'll get something else. We won't get 3-to-1, and we won't get 9-to-3 to 3-to-1. We'll get something in between. It turns out, with a lot of work, that turned out to be proof that crossing over occurred. So Mendel's work gave us three big important pieces of information. The first two were that genes were discrete chunks of stuff somewhere, and we could have different versions of them that were never combined together. We never actually take a gene apart and combine one allele with pieces from another allele. We get each allele individually in each individual and then hand it off to the next generation. That's particulate inheritance. And then he also found that different genes act completely independently from each other. So they're not in this situation. We would have the A gene for flower color here, and B, the gene for seed shape, let's say, on another chromosome. Not this pair of chromosomes, but another pair of chromosomes, and so on and so forth. That's the law of independent assortment. First is independent segregation, or the law of segregation. The second is the law of independent assortment. And then through some of the messy stuff that he couldn't explain, we came to realize later on by people following up on his work that it was evidence of this crossing over, something in between independent assortment and collective assortment. All right. So finally, a few other little ways in which genes express themselves. And one of the important ones, is this idea of epistasis, right? But that one gene interacts with a lot of other genes to create particular effects, particular characteristics. All right, so epistasis. The idea of epistasis is that there's one gene that can control another gene. And that second gene may have multiple alleles, and the other gene may have multiple alleles. So in this particular case, we're looking at the gene A, which controls coat color, and the dominant big A gives us agouti, which is this sort of mottled color, mixture. And lowercase a is going to be a solid color, like black or gray. All right, so this is the mottled, and this is the solid color right here. Then we've got a separate gene called C, which is responsible for making this, cells in the fur, or that produce fur, to make pigment. So there's a gene that induces cells to make pigment, not what color pigment. That's controlled by this gene right here, the A gene, but a gene that just says make pigment, the C gene. And in this particular case, there's a recessive version of it, an error of it, which causes cells to produce no pigment at all, regardless of what color they've inherited. And so here we have individuals that are homozygous for both of those characters. So homozygous for agouti, and homozygous for do they produce pigment at all, uppercase C times lowercase c. They're heterozygotes for all of these. So we do a dihybrid cross right here, but we get a really, really complicated and weird effect. Instead of the expected 1/16 being both black and not having any pigment right here, we end up with 25% of the individuals not making any pigment at all right here, and that's albino. The lack of producing albino is the lack of producing pigment is the albino thing, and it is a separate gene. It is not the gene that controls what the color is, agouti versus black right here, but in fact the gene that controls whether they do or do not have pigment at all. And so the color coat gene is epistatically controlled by another gene, in this case called the C gene, that determines how much pigment is produced. All right, we're going to go back and pick up the numerical analysis portion because you certainly need that. We will have a couple of test questions on that. I think I'm going to drop the pedigrees just a little bit too much. This is more important really as a student going forward. The probability basics right here, because this is going to apply to lots and lots of different things. So you know chance events, random things are described by probability. What's the chance of something happening, or the alternative chance, thing, or whatever it may be. And probability is generally denoted with a range of numbers somewhere between 0 and 1. And so 0.5 is the same thing as 50%. 1 is the same thing as 100%. So typically probability is written in these things, but you can convert it over into percentages. Some people like that better. A probability of 0, of course, means nothing in both cases. All right, and a probability of 1, which is equal to 100%, means it's guaranteed. This is the thing that will happen, so there's no chance involved at all. But we're only interested in things that have chance. So y'all all know this. If you flip a coin, the chance of it landing heads up is 50%. There's a 1 in 5 chance, 0.5%. Sorry, 1 in 5. A 1 in 2 chance that it'll be heads. And there's a 1 in 2 chance, of course, that it'll be tails. So 0.5 for either outcome right there. And, if you flip 4 coins, you might get all 4 heads, you might get all 4 tails, or you might get 3 heads and 1 tail. But if you flip 100 coins, you'll find that you start getting really, really close to 50% heads and 50% tails. If you flip 1,000 coins, it's going to even be closer, consistently. That's the way probability works. The bigger the sample, the more reliable probabilities are. All right. So first let's do what's called the multiplicative law of simple probability, and that's the chance of 2 things independent, rare things happening. So now we switch from coins to a 6-sided die. This is highly, imaginable for most people. So you know that the chance of rolling a5 a single time on a die is 1 in 6. What else could it be? There's 6 possibilities there. You get a 1 in 6 chance of getting the 5. And as we see right here, a different number, the chance of getting that different number just on a single die roll is still 1 in 6. Any number is 1 in 6. But what is the overall chance of rolling exactly a 5 on the first roll and then a 4 on the second roll? That's where you have to multiply their probabilities, and that is a 1/6 times a 1/6, which equals a 1 in 36 chance of getting a 5 followed by a 4. 1 in 36. Extremely, quite a bit lower than 1 in 6 right here. And that is the multiplicative law of probability. It'll always work that way. Same thing for the heads and tails question. We know that the probability of getting a heads or a tails is 50%. What's the chance of getting 2 heads in a row? Well, that's 0.5 times 0.5, which is 0.25. 50% times 50% is going to be 0.25, a quarter. All right. Now, the additive law, when there are multiple ways, as it says right here, for getting a particular outcome to occur, the chances of all of them together is added. You take the simple probability of the individual events and then add them all together. So So if here's the example. If one can win by rolling a 4 or a 5, we don't care which. We're not asking for 4 followed by 5 or 5 followed by 4, but just 4 or 5. We know the chance of rolling a 4 is a 1 in 6. We know the chance of rolling a 5 is also a 1 in 6. Well, now we add them together. We add them. So the chances of getting a 4 or 5 is actually going to be greater than getting either individually, right? So 1/6 plus 1/6 equals 2/6. That's the way the math works. And then if you simplify that, it's 1 in 3. But we could use 2/6 just to keep it kind of familiar right here. So that's, if we want one of several possibilities. So, I want heads or I want tails. I want 2 heads or I want 2 tails. Each of those will work on my coin flipping. Well, we know the probability of getting 2 heads in a row is 0.25. And the odds of getting 2 tails in a row is also 0.25. So the odds of getting either 2 tails or 2 heads is 0.25 plus 0.25 or 0.5. So applying it to that sort of problem right there. Try something yourself. And then we can combine them together for more complex situations. All right. So here we're looking at the chance of rolling a 5 and a 4 in any order. 5 and 4 in any order. So we remember chance of 5 is 1 in 6. Chance of 4 is 1 in 6. So the combination of a 5 first and a 4 second is 1 in 36. We know that already. But for the other possibility, in any order right here, we're going to get 1 in 36 for having a 4 followed by a 5. And then if we want to add those two combined together, 1 in 36 plus 1 in 36 equals 2 in 36, or 1 in 18, a higher probability. Well, that's just what I did previously with my coin toss. The chances of getting 2 heads in a row is 0.5 times 0.5, or 0.25, because you multiply them. The chances of getting 2 tails in a row is 0.5 times 0.5, which equals 0.25. The chances of getting 2 tails or 2 heads is 0.25 plus 0.25, or 0.5 again. All right, so that's a little bit about probability, and these are useful because you can do this sort of thing right here, no matter how big a problem you want to look at, how many different characters at the same time, knowing your outcomes for particular individual events, and then combining them all together for a particular combo. We're not going to try and run through all of them, but to come up with any particular combination, we'll either use the multiplicative rule or the additive rule. So for these particular problems right here, for one of these particular outcomes, it's going to be the multiplicative rule. So let's go ahead and take a look just on the calculator doing this in probabilities. For the possibility of getting a green round dwarf plant, and we know green and dwarf are recessive and round is going to be dominant. So let's get the calculator over here. If I can get it, come on, there we go. All right, so green, 0.25, times round, 0.75, times dwarf, 0.25, equals this, 0.04. It was 0.0468. Well, how many out of the 64 plants that we were looking at in that particular tri-hybrid cross back there? Well, let's multiply this times 64, and we get 3. And so 3 is the answer right there. That's just going ahead and for a given population. And we ask, what are the probabilities of getting a green round dwarf plant? And there are, in fact, 3 possible. 3 out of 64 getting this. How do I know it's 64? Well, I could just add these all together. 27 plus 9 plus 9 plus 3 plus 9 plus 3 plus 3 plus 1. Or I could do, well, this is 4 times 4 times 4. 4 times 4 times 4 is 64. So use math at some point. This is good, but this starts getting a little bit laborious. And a 4 hybrid cross, would have to have another row down here. It'd get even bigger and bigger. Sooner or later, we just want to switch over to using math to answer these sorts of questions right here. And if we wanted to answer a question about, what's the probability of either getting a green round dwarf plant or a yellow round dwarf plant, we just run through the combinations for both of those and then add them together. The chances of either getting that yellow round dwarf plant or a green round dwarf plant would be the probability of getting a green round dwarf plant, which is what did we say? 0.046. And the chances of getting a yellow round dwarf plant is going to be a bigger number, and then we combine those together and it will turn out to be 9 plus 3 out of 64, or whatever that is. So you will get some testing on, where is it? On using the multiplicative law, the additive law, and then combining the two together. All right, and that concludes this chapter and really the bulk of the material for the last unit, which will be included on the final exam.